Thank you for the kind introduction and for the opportunity to tell you a bit about what we are doing in our effort to get an antiviral for COVID-19. Now, since we're all living through this pandemic, I'm going to skip the customary slide of how bad the disease is and just jump straight into the science. Now, as you probably know by now, COVID-19 is caused by the virus SARS-CoV-2. And what I have on this picture is just a representation of when the virus has infected a human host. Its RNA gets translated into this long polyprotein, which needs to be chopped up into the different viral proteins for all the downstream processes. And in SARS-CoV-2, there are two cysteine proteases that does this job. The one is PL-Pro, and the other one is the main protease M-Pro. And you can see we've just color-coded in pink all PL-Pros cut sites, and then in purple all M-Pros cut sites. Now, the COVID Moonshot project, we are very much focused on the second protease M-Pro. Now, just to give you a bit of a timeline of how fast everything has been moving this year. So the structure of M-Pro was solved and deposited by a group in Shanghai in end of January already. They contacted Diamond, and Diamond managed to clone and produce the enzyme on site in Oxford by mid-February, Valentine's Day, actually. And shortly after that, we had our first crystal structure at Diamond. The group in Weizmann, being led by Near London, managed to do a covalent fragment screen. And in Oxford, we did a crystallography fragment screen of about 1,500 crystals. And then by the 18th of March, we had our first 78 fragment-bound structures that we released to the web. And our release was more or less with a web page. We didn't follow the traditional route of depositing to the PDB or publishing a manuscript. And the reason why we didn't initially do that is because we wanted to get the data out there to the public as fast as possible so people can start using it as fast as possible. We have by now deposited all these structures to the PDB, and the article's already been released. So if I just go back to the data of the fragment screen, I don't know how familiar you are with crystallography, but this gray thing here is just a representation of the enzyme, and all this colorful sticks in here is all the different fragments that bound to the enzyme. And what we've done is we just divided the active site into 4 sections, so as you can see here. And all I really want you to notice at this moment is that we have fragments in all 4 of the sections of the active site, number 1. And the second thing I want you to notice is that if you look in it, there is definitely fragments that overlap with each other. So from a data point of view, this is beautiful, but the question is, so what? What do we actually do with this, and how is this going to help society as a whole? And this is where we get to what was called Project Moonshot. A few of the academics got together and looking at this data set, but we have to be able to do something with this. Somewhere in the merge space, there should be a molecule that we can easily make that will be a potent inhibitor of this enzyme and hopefully being a safe antiviral. And I've been told the conversation went more or less like this, when near London at the Weizmann Institute in Israel said, but let's crowdsource ideas. Let's ask Derek Lowe, who's got a blog on drug discovery, if he will publish it and ask for smiles, and maybe we can collate it all in a Google Doc. Fortunately, at that stage, Alpha Lee, who works at Cambridge instead of PostEra, said, but they've got a website that they could use and for the submissions. Of course, if we've got structures, John Kandera came in and said, but he's got all the tools in place to rank the submissions. And if we can rank them, then we'll know what to make and not waste time unnecessarily. And then if we know what to make, then Frank van Delft at Oxford said, but we've got resources at Diamond and at Oxford to test them. So what happened is they pulled their resources, they pulled their grant funds, and the project got born. And as we advertise it, this is the ambitious mission to produce a COVID antiviral, hence the name Moonshot. Now, how did we went about it? We basically had this website built by PostEra. We used social media to get the word out there and made basically a submission form where we told the med chemists and all the chemists out there, is like, listen, here's the crystal data, here's the fragment data, see what you can do and suggest molecules that we could make that you will think will be active. You can do your submission by smiles, you can do your submission by drawing the structure. The only thing we ask you is that you will let us know how you decided to make this molecule. So what crystal structures did you use? What calculations did you use? And this is basically just a way of checking what was the rationale and what is the feasibility of this. And this then went into a normal type of three-pronged cascade. We design molecules, we synthesize the molecules, we test the molecules, and we repeat the process. Now, this is also where CDD came into our lives, and we can see we made 3 projects in our CDD Vault. A virtual project where we capture all our design molecular ideas, a synthesis project for all the molecules that we decide we're going to synthesize, and then a made project for all the compounds that goes out to be tested. On the one side, we very much have a med chemist and a community that helps with designs. And on the other side of the equation, we have Inamin, who is in charge of all the synthesis for us. Now, of course, nothing is ever as easy as shown in a picture like this. So somewhere between the community and design phase, there is a lot of fact-checking that needs to be done. We need to check for duplicates in submission. We need to check for synthetic feasibility. After we've identified the molecules that we think might be good, we have to some way prioritize them and do various analysis and docking and scoring methods to decide which ones should actually be made first and might have the highest chance of succeeding. As I said, Inamin does all our synthesis for us and handles all the synthesis records and pathways routes. They are also our inventory, so they keep stocks of all our compounds for us and send it out to the labs that we needed that. And on the other hand, we also use other CROs to synthesize some of the compounds, and all those CROs send their compounds to Inamin, who then handles everything for us as a central deposit place. And then, of course, once the compounds get sent out to be tested, we need to make sure that the files make sense, that we send out all the right plate maps. Another thing that happens is that once Inamin ships us compounds, we don't necessarily know the stereochemistry yet. So there's also an iterative process of going back and fixing things like stereochemistry. And then we test the compounds, we upload the data to CDD, we analyze a lot of this data on CDD. And then the process comes of pulling the data back into a public forum. So we use a lot of PostEra for that, and the CDD API that Charlie mentioned earlier. And then we broadcast the data. Since this is an open source project, we really want people to know what's going on. So there's discussion forums and blogs in which everything gets disseminated. I just want to highlight 3 tools here. The one is the Fragalysis tool, in which we use for crystallography, and I'll get to that again later. The other one is PostEra, and PostEra is really key and involved in managing and doing most of the stuff on the top end of the slide. And then we use GitHub also as a data depository for all our raw data files. But since I am at assay level and I do most of this work with CDD and our assay data, what I'm going to focus on in the rest of my talk is just this little test circle in the bottom. So the next slide is a bit busy. So don't worry about all the details. I'll point out what I want to say, but this is just an overview of our current assay cascade, going from our synthesized compound to hopefully what we want, a preclinical candidate in the bottom. So the first level of assays is really easy. We receive the compounds at Oxford and at the Weizmann Institute, and we send all of these compounds through to X-ray crystallography, to FRET assays, which is a biochemical assay, and then our MS-based biochemical assay. Each one of these blocks represent only one laboratory. They do a set of compounds every single week. We get the data back in exactly the same format. To be quite honest, the Weizmann lab uploads it directly to CDD, so that's even easier. And we know exactly what we are looking for. So there's a very defined criteria that says Are we progressing this compound, or is this the end of this compound? And at this level, it is still very much one of us can pull a report from our CDD data, make a decision, and progress it. Now, the further down the line we get into the cascade, things get a bit more murky. One of the main reasons for this is that since we're a community-sourced open source project, we do not necessarily use specific CROs or specific labs for all of the assays downstream. And what happened, especially with the viral and the cytotox assays, is that we had labs approaching us, or we had friends that we approached and asked if they were willing to test a certain amount of compounds for us. So we literally have viral labs that said, yes, I can test 8 compounds for you, or yes, we can test 1 plate of compounds for you. The problem with this is that not all viral assays are the same. There is a wide range of different readouts, different methodologies to do it. And that also means there's a wide range of types of data and data interpretations we get back. From a data point of view, from what I'm doing, it's because we know we're reliant on their goodwill, and because we know they offered the services for us free, can't go back and dictate. And it's like, oh no, but if you want to test 8 compounds for us, you need to use CDD. You need to do it in this way. You need to give me this type of upload file. I mean, that will be very unfair and very ungrateful from me, to say the least. The other thing which makes this more complicated is that as soon as you have a lot of different data systems, it gets a lot more difficult to define the criteria specifically of what is interesting and what is not interesting, or what might potentially be a good compound and what would not. And the fortunate position we are in the COVID Moonshot is that we have the expertise of ex-pharma people, we have expertise of a lot of med chemists and people who have actually taken drugs to market that can look at the data, look at the complete whole picture and advise us of what to do and how to proceed. And in a big case, a lot of this is a learning curve for all of us on the project, but it's really, really interesting and good. So what I'm going to do now is just show you an example of one of the viral data sets we got back. And with the link, and it's going to be a bit of a CDD ad, but just how CDD is actually making our lives a lot easier. So this is a assay plate of a viral assay. And this is actually really beautiful and a really rich data source. Now, if you look at these red columns on the outside, this is the cells, the A549 cells, that has an mCherry reporter. So this is what live cells look like. When it is green, I know this probably looks yellow on your screen, that means that is the color of the virus. So the virus has got a GFP reporter in it. And if you just look at all the inside where we've tested compounds, when you see red and not yellow, like in where I'm showing my mouse, that means the virus died. So that's a good thing. That means our compounds are working, the virus is dying. If you ever see black, that means our compound is also killing the cell strain that we're testing in. So that's a sign of cytotoxicity. Now, the good thing with mCherry and GFP promoters is you can put it on a spec and you can get a readout. And when we uploaded this to CDD, we were actually which we won't see visually from here, but we are actually managing to draw very nice EC50 curves for our antiviral data. And the same, if we just look at the mCherry readouts, we can do the same and we can get very beautiful curves for the cytotox data. Now, why this is so exciting is, firstly, because we get a lot of data from a plate which you don't visually can see. But secondly, we're getting a uniform output, which makes it a lot easier for all our collaborators or all the people who have some interest in it, or the community that wants to come back, to look at the data and say, oh yes, I know this type of plot, this is what this means. This is moving on a bit from the data, and I'm just actually going to tell you a few of the lessons we learned along the way and things we had to start implementing to make our vault a lot more easier for people from outside to use. Also taking into account we're a group from a lot of different fields. So I'm from very much a wet lab science experience, but we do have our chemists, we have our industry people, and we've got our clinicians. And not all of them are used to the same data or looking at the same data in the same way. And one of the things we actually realized, there is sometimes a lot of this big discrepancy of just how we understand each other. So the one thing we've now implemented is naming conventions. So if we just look at our protocols, we have decided that all our protocols need to start off with what type of assay is it? Is it an ATME assay? Is it an antiviral assay? Is it a cytotoxicity assay? Is it an enzymatic assay? So that just groups it together and makes it immediately clear what we're looking at. We didn't follow this up by just a description of the assay. So if I look at the ATME one, it will say it's a metabolic stability assay. I'll get back to the antivirals now. Well, yeah, so getting to antivirals and to the cytotox, the first thing that's important is what cell line did we use. So you'll see we'll next thing say it's a calorie-free cell line. It was tested in Vero6. It was tested in A549. And for these ones, we followed up with what was the assay type. So was it the FFU assay? Was it the plaque assay? Was it the imaging assay up here? And then lastly, what we list is the laboratory in which the assay was done. So I'm going to show you an example later why this makes it life very easy. And Charlie also mentioned collections. So we've actually realized that collections are a really powerful way of just keeping our data together. So we employing more or less the same type of naming convention, but we're using collections for our different batches that we send off to our different collaborating labs. So if I just use the antiviral again as an example, we will say that we have an antiviral shipment of our second batch of compounds that we're testing that we're going to send to Oxford, and we've decided to do it on this date. So going forward now, if I need to update the tracker, I can easily go to the collection, pull the data, update my tracker. If our med chemists need to update us on our Wednesday meeting about what's being done, he can go there, pull the data, update his presentation. The same if our clinician wants to see what we've covered and what we haven't covered, she can go in, pull the data, and very easily see, okay, listen, we've assayed these ones, but we haven't assayed those ones, here's the problem. Another thing is because we do most of the data uploads ourselves, there's only one lab outside in our collaborating environment that actually use CDD for data uploads. We've realized that we have the potential of losing a lot of the info that you normally get in ELN pages and protocols and metadata. So what we started to implement was to use the ELN pages, firstly, to collect all the protocols with, but also further, because most of the assays that we, groups that we work with always go through the whole process of writing a report and showing what your water analysis is. As a dumping, so we basically use as a place where we can dump their regional reports and their original data. Which makes this really powerful is that if anyone ever wants to go back and query a result, they can go back to what the original lab said. And this is kind of a source document that just verifies our data integrity. And another thing we started to do was sometimes you just get so excited about any type of data that there will be this whole long email thread that will get lost somewhere. So just some of these discussions, just also starting to dump them into an ELN page. Now going forward, this might look like a lot of work, but we know that we are not just building it, trying to get one molecule that goes into a clinic. We are also collecting, and very much aware of it, a database that can be used for machine learning protocols, or that can be mined further to look at correlation between assays. So all of this data would actually be very handy to have there as a backup and for the completeness of our database for any of the downstream other, so not the drug discovery point of view, source uses we want to use it for. And then another thing we started to do is we started doing compound tracking in the Vault. So I spoke to Charlie about this, and he actually suggested it just a while ago, is that we can track what assays or compounds go to by just using a normal protocol, but defining a parameter called assay type and making that a protocol condition. So this makes it easy if I want to go back and see which of our compounds have gone into antiviral assays. I can just run a search on our assay request logistics, choose my assay type as antiviral, and run the search. And just to show you what our the type of data upload looks like. We have our protocol defined, in which we define our assay type, which laboratory we asked to do the assay. So as I said, for antiviral and cytotox, we have numerous laboratories doing this. When did we request the assay? When was it completed? And who was the person who requested the assay? And the last thing we have here is a progress indication. So for most biological assays, we have a very set outcome. We know it's active or it's inactive. When we get to some of our other outputs like crystallography, the endpoint is never really that clearly defined. And sometimes it can just be when we have tried everything we could and we can't think of something else. So in that case, a progress bar is actually really great because we can say, yes, we have a crystal structure of a compound. No, we do not have a crystal structure, but we're still trying to get one. Or no, we have stopped now because we don't know what we have done, exhausted all our resources to do this. And then another thing we actually also started misusing a lot more for our compound tracking was just the batch fields. So the first thing I want to show you is that we get a shipment of compounds from Enamine every week, and we number these compound shipments with the by shipment numbers, so shipment 1 to 31 we're up to at this moment, and by the date they were shipped. And we use this information for all our first line of assays. So we will say, we send you shipment 30, please assay this. We also use it on the crystallography side, is shipment 30 has gone into crystallography. On the other hand, our plates are numbered with the shipment names. So if we ever need to go back to a compound, we can pull the data of what shipment it was, know when it went to crystallography, know when it went for basic assays, but we'll also know, okay, this is this plate in the -80 that we need to go look for. Just linking and making our life easy, Enamine generates catalog numbers and batch IDs. So we started uploading that for CDD also. So this just always makes it easier to link back from CDD to Enamine with exactly the compound that we want. On the GitHub side, we always have a lot of CSV files that gets generated in the whole process of making plate maps, of fixing the enantiomers, of naming. And we decided on CDD to actually have 2 fields where we just list all the file names that are in GitHub that is related to a specific compound. And this just, once again, just gives that easy link back to where do we have to go look for information and where do we find it. And then the last thing, what our medchemist started doing in the last while, so we've got discussion links on PostEra and a lot of design rationale that's recorded. And we are now also linking this into our batch fields for our compounds. So if anyone is wondering about anything, you can go to the batch field, see what is the PostEra link you need to follow, and get the data from there. And very soon we'll also be able to link our crystal data like that. So this is basically what all this type of innovation have done is. So you remember I told you about we had specific naming conventions. And the reason we did do this is this report that I'm showing here was pulled by one of our clinician. And one of our collaborators wanted to know how is her series of compounds behaving, through what test did we put them, because she had an NIH grant and she needed to upgrade data in NIH grant. So the person could literally go, put in the series, pull all the data, and just from what we get on the CDD report, know, okay, this is the assays that is gone into, very specific. These are assays the compounds didn't go in through. This is the overlap, but also could generate a very nice report with all the IC50s and percentage inhibition values needed. So we went from having a lot of different type of viral data with different outputs to having a uniform look that anyone could understand if they quickly look at this. So this is of course the question is, so where are we today? So I'm just shifting gears a bit. So since February to where we're now in November, we have built an international group of collaborators spanning 3 continents. We had a GoFundMe campaign where we have managed to raise just over £43,000. And on the compound side, which is of course the important one, we had just over 14,300 molecules submitted to the PostEra site, of which just under 1,300 was synthesized and tested, and we have 211 new crystal structures with compounds bound to them. Now, what I want to show you now is just how we basically report data back to communities. So this is the PostEra website. We pull the data from CDD with the API to PostEra site, where it gets plotted. You can see we've got our compounds with the names, and we've got search boxes. So if you go search for the compounds, you'll see all the data that gets generated on CDD gets reported. So you can easily go as the community and go see what is the IC50s inhibition, but you can also go and see a bit more of the rationale and a bit more of the details on the molecules as we've recorded it. Another tool I want to show you, there it goes, is, let me just pause quickly, is our crystallography tool, Fragalysis. So we have now, in the last 2 weeks, managed to make hyperlinks that's specific for each of the compounds on this Fragalysis view. So this was just once again going through the PostEra website. If you go find your favorite compound, you click on the view on Fragalysis, it will open this browser. So I'm just showing you this. This is our viewing of crystallography data browser. This thing you see here is the enzyme, and you'll soon see popping up here. It will tell you all the different sites where we have compounds, and we'll list on this side all the different compounds that is bound in this crystal structure. Yeah, there it pops up. And then it shows you in a 3D way how a compound is bound, what interactions formed. And as you can see just from moving around, this is interactive, so you can turn it to whatever way you need to be. Now, both PostEra and both Fragalysis, there is a lot more to say, and I won't do I'm not doing anything justice to it now. But if you're interested, please contact us. We're more than happy to talk about this. And then the last thing I want to end up with, I just want to give you an idea of the process. So if you were a user out there and you've submitted a molecule, what would have happened? So this molecule here was submitted just as the start of the Moonshot project. It was a contributor, Trifan Saganis, and Oxford University. And you can see what we immediately do is we give the compound a name that is linked back to its designer. So TRA being the first 3 letters of his name. And he listed himself as being from University of Oxford. So when it pulled in his origin, it pulled in UNI as where he was from. And when he submitted this molecule, he gave us a rationale saying that it was based on these fragments in the original fragment screen. And it shows us some of these fragments. And then just from the PostEra side, you can see we've annotated and says this is a free aminopyridine-like molecule. Of course, it's now a bit down the line, so we already know it's assayed. So what happened to this molecule? Well, this molecule, as I said, was submitted on 17th of March. It took us a whole 57 days to go from when it was submitted to having final data assayed out, which is fine. This was our first round of doing all of this. Everything was still being in the process of being set up, being optimized, growing pains, basically. And when we assayed it, it had a mediocre IC50 of 24 micromolars. Not that great. But as the design process got on, we started building and changing this molecule. And we got to where we are today, where we've got this benzopyridine with an IC50 of 100 to 14 nanomolars. So all of a sudden, this is a lot more promising than our original hit. And this is also now the scaffold that we do using for our lead series. The other thing you will notice is our turnaround time was basically almost a third of what it was in the beginning. So I know this is not a drug, but this is the molecule that we're basically doing our SAR on, and which we are sending now for initial viral and acne profiling. And I think all I can tell you is watch this space, and with space, we'll probably refer you back to the PostEra website. There might be something interesting coming out. And with that, I just need to thank everyone. So the Moonshot consortium is quite a big consortium, and in an effort for me not to leave out any names, I just pasted in the author list from the bioarchive paper we have on this process. And then, of course, I want to say a specific thank you to Mariana, Susanna, and Charlie, who was really awesome in helping us set up our CDD Vault, in supporting us all the way through, and is still on a regular basis answering questions for us on all of this. And with that, I'll hand back over.